So I'm Peter Piot, and I'm the director of the London School of Hygiene and Tropical Medicine in London, and where I'm also a professor of global health, and by training I'm a physician, infectious disease specialist, and I have a PhD in microbiology. And when I was in my last year in medical school, long time ago, I'm an old man, and that was in Belgium. I'm originally from Belgium. That's my accent, is a Flemish accent. I went for what today is called career counseling, a term that didn't even exist then. And I asked my professors, some advice, and I was very interested in epidemics and in working in Africa and problems, health problems in developing countries and so on. And the verdict of my professors, except the professor of microbiology, of course, was here. This was what they told me: no future in infectious diseases. This was in 1974. And don't we have antibiotics? Don't we have vaccines, even if we didn't have any of the vaccines that you heard about before? And don't we have hygiene? And of course, these were very, very important breakthroughs in the fight against infectious diseases. But I always thought that, one, you have to follow your passion. When you don't overplan your career, do what you really like and what you're really interested in, and that's what you will do well. And also, I'm slightly stubborn, so I went for infectious diseases. And then 2 years later, this, not exactly this, but a thermos like this arrived in the laboratory where I was in training in clinical microbiology. That was part of the training in infectious diseases. And a thermos, a shiny blue Chinese thermos, arrived straight from Kinshasa. It was a pilot from what's now called Brussels Airlines, had deposited this thermos at the reception of the Institute of Tropical Medicine in Antwerp, where I was then working. And with a little note, from that this was from a patient who had died with an unknown diagnosis, hemorrhagic fever, yellow fever, ? And we were equipped in that institute to work on arboviruses. These are viruses that are transmitted by all kinds of arthropods, mosquitoes mostly, but ticks and all that. And yellow fever is one of them. So what we did was we did the routine type of ways of isolating a virus. And today we've got very sophisticated tests. We go straight for the genetic material, RNA or DNA of the virus, through tests called like PCR and so on. But in these days, it was like inoculating cells, that are in tubes or in flasks, and inoculating mice and newborn mice and guinea pigs and so on. Fairly artisanal type of science, I must say. And that's how we isolated viruses. And then, to our big surprise, a virus came out of it that's like here. And in general, viruses are spheres, are round, or are look more like crystals and so on. There are only very few viruses that are like this, very like worms-like structures. And these days, since 76, you weren't born yet, there was not only no mobile phone, if you believe that, no internet, no fax. And so when we saw this structure under the electron microscope, we said, we had to go to the library. You know what that is, a library? Yes? And look into an atlas of viruses. And there was one virus that looked exactly like this one, and that was called Marburg virus. Marburg is a town in Germany where several years before a similar virus, a cousin actually, as we know today, of Ebola virus, had caused a deadly epidemic among workers at a vaccine preparing plant, a vaccine plant, and also in the former Yugoslavia in Belgrade. And it was a virus that came from infected cells, monkey kidney cells that were used to prepare polio vaccine. So we thought, this may be Marburg virus, which created a bit of panic because it's so highly lethal and we were not equipped with, to work with viruses like that. That is only allowed to do in so-called category 4 or high containment laboratories. So what we did then is we sent the sample by commercial flight to Atlanta and to Portendown, but to Atlanta, that's where they were first. And why Atlanta? Because there at the Centers for Disease Control, they have this special laboratory. And they could demonstrate that this was not Marburg because it did not react with antiserum, antibodies made against the Marburg virus. And so it did not, the new virus did not react with it. And this is why we could then say that this is a new virus. And that was published years ago in The Lancet. The two papers, one from our group in Antwerp and then from the Centers for Disease Control. And so that's how, that was the beginning of my career in global health. And the viruses need a name. Sometimes it's given after, the name is given after the person who discovered it, but that's really considered bad taste. And then a very common name giving comes from where the virus was isolated, like Marburg, or where the disease was found, Lassa fever. But we thought that could be very stigmatizing. Just imagine when you introduce yourself and say, hi, I'm from Lassa. It's not a great introduction. And so we said, let's go for a river that is close to where in the meantime, we know was an epidemic. And this is the Ebola River in northern Congo. Here you see the virus, and that's the river. It's a river that ends up in the end in the Ubangi and then the Congo River. And that's the origin of the name of the virus. Now, by some extraordinary coincidence, at the same time as there was an epidemic, so this virus came from Kinshasa, the capital of Zaire, where this patient had died, and I'll tell about that patient. But there was an epidemic going on in northern, what was then called Zaire, now the Democratic Republic of Congo, around a small village and a mission post called Yambuku, near the town of Bumba. This is the Congo River, which is the second largest river in terms of volume after the Amazon. But in some extraordinary coincidence, there was a similar outbreak was happening in Sudan, now South Sudan, around the towns of Maridi and Zara. And so what happened then is that a few weeks after having isolated this virus, I went to Zaire. I had never been to Africa. I had no expertise, but many people who were far more experienced than I was had maybe second thoughts before going there, because the rumor was that this was a very deadly epidemic. And there was another team that went to Sudan, and a US UK team. And today we know that these were epidemics caused by different viruses. And the virus that caused the outbreak in Zaire, in Congo, is still called the Zaire Ebola virus, you see here. And that's the virus that has caused most outbreaks and is the most lethal, has the highest mortality rate. And then we had the Sudan virus, which has a mortality rate of about, that's 50%, around 50% only, as compared to in the original outbreaks, 70 to 90% for Ebola virus. And then subsequently, Bundibugyo virus, this is a place in Uganda, was isolated. Thai forest virus, only one case in Côte d'Ivoire. And we have Reston virus, but that's an Ebola virus that doesn't affect humans, only certain primates. And that came from the Philippines. But Reston is a town in Virginia, or is Maryland? Yeah, so around Washington, where you have a primate colony. And some of you may have seen The Hot Zone, a film that's about this Reston virus. So a lot of the film is not really factually correct because it doesn't really kill people. So I went to the epidemic zone, as we call it. This is the picture of this is a Catholic mission in northern Congo, and where you had a nuns' convent, and then a father's convent, and a church, and a school, and a hospital, and a cooperative, you know, the classic constellation of things. And what had happened there, you can see here, and this is how the world got to know it, that 4 nuns, 4 missionaries, had died from this mysterious disease, and 1 father, 1 priest. And we are really in the equatorial forest, where people live in very close contact with the forest. So this is the road, and you see a village here and a village there, people living here. They have the fields down, they hunt and so on. So there's intense contact between people and animals and whatever is there in the forest. This was the team that came, that arrived in Yambuku, and it was an international team, of course, with Congolese colleagues. This is Joel Bremen from the Centers for Disease Control in Atlanta, as Pierre Sirot from the Institut Pasteur in Paris, and then this is when I was a bit younger also. But I'm showing this also because the sisters had read, this is in a French book about epidemics, what to do, that in case of an epidemic, you have to establish a cordon sanitaire, a sanitary cordon. I mean, and they had, a cordon is a cord, you know, and they had interpreted that in a literal way and put a string around their guest house where they were waiting for their death. And had said, there was in Lingala, the local language, there was a message and say, don't cross this barrier. Anybody who will cross this barrier will die just like we will. And so we arrived there, and the nuns were telling the story of an epidemic that was still going on. So we had, as a mission, several things, and that's what I'd like to tell about. How do you investigate an epidemic of unknown origin or unknown transmission? And first of all, we had to see, are there still active cases? Is this still going on, or are we arriving when it's over? And also collect biological material to confirm that this was exactly the same virus would come out of it as we had isolated from this nun who came from Yambuku and went to Kinshasa for medical care. And so we saw several patients in the villages, and many were terminal, all of them died nearly, and nearly all of them died, and there was hemorrhagic signs. What does that mean? It means that, you know, there's blood coming from all kinds of orifices. And here, this is the clinical picture that we could describe. So Ebola virus hemorrhagic fever starts as basically the flu. You know, you have some fever, headache, myalgia, so muscle pain, and some joint pain, abdominal pain, sore throat. So all of us have had that, I don't know how many times. And just don't think that you have Ebola next time you get this kind of things. You know, this is this is how most viral illnesses start in terms of clinical expression. And but after a couple of days, 3 days, things deteriorate in many patients. And what you see here is nausea, vomiting, diarrhea, conjunctivitis, and then in a number of patients, about 25 to 40%, depending on the outbreak, bleeding. It can be rectal bleeding from the eyes, from the ears, vaginal bleeding, and so on. So all that. So that's the clinical picture of Ebola. Now, in the meantime, we were aware that this was probably an infection with a high mortality. I say probably because when you have not studied this infection, it could be that there are many mild cases of this infection. That's not unusual. You know, many, like in influenza, many people are infected during an epidemic, millions, but only thousands are dying. I mean, far too many. So there's a whole spectrum of clinical expression of the natural history, and we didn't know. And we still don't know exactly what's the percentage of people who are infected really develop this serious disease. But in any case, we didn't want to take the risk, and this is how we protected ourselves. This is what the uniform is today. We had simple latex gloves, a paper gown, a paper mask, and motorbike goggles. That's because, you know, and why do you do that? The most common ways of entry of a virus, the most vulnerable parts of your body are your hands often, because, you know, the skin may not be intact and so on. You can come in contact with blood and so on, because we were drawing blood, for example. And it's the conjunctiva, it's the nose, and it's the mouth. So that's what you really want to protect. Although we didn't know, maybe is this mosquito-borne, is this airborne, and so on. We don't know. So we had really very basic protection. I remember we had received a number of kind of gas masks, but I tried it once and couldn't breathe, and this is also where it's extremely hot and humid. We had a person who developed fever and diarrhea and so on during this outbreak and was evacuated. Fortunately, he was not infected. However, here in Portendown, where were the army laboratories, military laboratories, and where they were working with Ebola, then one laboratory worker, laboratory technician, became infected, but fortunately survived. So that's really part of the risk. Now, when you are confronted with an epidemic of unknown transmission or with any epidemic, you start by asking three very basic questions. And the keywords are time, place, and person. So you try to define that in a quick and dirty way. In a couple of days, 3 days, you try to answer these three questions: time, place, and person. And the first thing you do is to draw what's called an epidemiological curve or an epidemic curve. Here the number of cases and with the date of onset. Here you've got the date. And so this epidemic had started actually the very end of August. And we came in the 18th, so here. So that's the right time for an epidemiologist to arrive, because if you come here, then you'll be fired when the peak is there, because you said you're incompetent, you didn't have any impact. When you arrive here, they say, oh, it's thanks to you that the cases go down, because this is onset of disease. So in other words, people die after 1 week to 2 weeks. So it's not when we arrived that there were hardly any cases, because many of these people were still alive but would die. And so this shape of the curve told us already a story. It told the story that it was an epidemic of fairly recent onset, that it had developed very rapidly. In about 4 or 5 weeks, it had reached a peak, and then suddenly also it went down. And so we asked, this is the time question, what happened here? Why, was there anything that people could point to as that may have influenced this decline? And the answer was that around this time, people started understanding that there was something wrong at the hospital. So the hospital was abandoned, was closed, and retrospectively, that was a major factor in the control of the epidemic. It was nothing else than that. Secondly, place. Now, again, this is a time when there were no laptops and computers and so on, and I still have my original notes of every case and so on from '76, and gave that to a postdoc in June or May, and for further analysis for mathematical modeling and so on. And he asked me, so where is the spreadsheet? I said, this is the spreadsheet. You know, it was made by hand, and so the second question then is place. And place was here. There were no maps then, or very old maps, and the mission was here. This is the Congo River, etc. But what we found is that the closer you would live near this mission hospital, the higher the risk, the higher the number, larger the number of cases per village of this hemorrhagic fever. Again, pointing to the fact that there's something going on at that hospital. And then thirdly, most importantly, is person. Who gets this? And you do, again, something very simple. You divide it into age and sex, the cases. And so here you see age distribution. You put it in several categories. Newborns and infants, 1 to 14, 15 to 29, 30 to 49, and over 50. There are not that many old people, very old people in that part of the world. And then in blue, you see men or boys and girls and men and women. And this really was the key. This provided the clue to our understanding of what was going on. First of all, when you see that there were relatively few children, actually quite disproportionate number of newborns and infants, and that really already went against transmission by mosquitoes or other insects. Because why would an insect not bite to the same level children? And actually, usually it's the opposite. Children are more affected. And secondly, the major difference was here. You see there were about double the number of women than men between 15 and 29. Now, the first question you have to ask, maybe that's the normal distribution. It could be, and that's the case in some parts of the world where men of working age, they are not in their villages, they are somewhere working in the mines or in the city and so on. But that was definitely not the case here. And since we were a bunch of men, it took us several hours to see and to understand what the difference is between men and women between 15 and 29. I'm sure the women here know immediately what it is. Women can get pregnant at that age. And indeed, some of us, I don't remember who, had the idea that this was the case. And then we went back to the forms, and the question was, is this excess of women, is this indeed, can be attributed to pregnant women, to pregnancy? So we looked at the cases, the inventory we made from going to the villages, and also compared it to the data from the hospital, because there was an antenatal clinic. And indeed, basically, the excess of women was entirely due to pregnant women. So then the question was, but what on earth? Why would pregnant women be more vulnerable? Is this a biological factor because there's immune suppressed, or you could, all kinds of possibilities, or something else? And so so all this we knew in about 72 hours. But then what took us much longer was the why. And that, you'll understand why that was difficult. So in the end, we found out that indeed, most of these women had come to the antenatal clinic of the hospital, which is a good thing, because delivering at home in that environment is a risk factor for maternal mortality, for newborn mortality. So the women went there, and then we wondered, what is going on at that antenatal clinic? And the answer was then, in the end, we found is that Mother Superior would distribute 5 syringes and needles every morning, give that to the outpatient department, and everybody who would come to the consultation, to the antenatal clinic also, would receive an injection with vitamins, calcium gluconate, frankly, fairly useless things from a medical perspective, but it was very popular. And these were not disposable syringes or needles, but they were reutilized. And at the best flushed in water, sterile water, hopefully. And so in other words, this is one of the most efficient ways of transmitting viruses from one person to another. Not only Ebola, but hepatitis. There are huge epidemics of that. You could even have HIV, as we have an epidemic in Cambodia at the moment, and so on. So this is the story how we found out that the first mode of transmission, and that was needles and syringes. Secondly, we saw another thing in terms of the WHO, and that is there were, this is from another epidemic, but that in of the 17 people who worked in the hospital, it's a 100-bed hospital, that 11 of 17 had died, healthcare workers. And this is from another outbreak in a place called Kikwit, also in Zaire, but then much more to the south, and where you see a whole chain of infections and of deaths, all in hospital workers, a laboratory technician, an anesthesiologist, a nurse, again, anesthesiologist, and 3 doctors, and 3 patients in the hospital. So that was also very, very special. And we didn't think that all of them would have been infected through, let's say, accidental needle pricks. What has happened and what we're seeing also in West Africa today, over 500 healthcare workers have died from Ebola in West Africa today. 500 out of total death toll is now moving to about 10,000. So let's say roughly 5–6%. That's enormous. That's far higher than the percentage of healthcare workers in the general population. So that was another factor. So we thought caring for a patient, treating them, is a very high risk factor. And then the third element which came was that people told us that 1 week after a funeral, that many family members would develop this disease and would subsequently die. Family members and people who went to the funeral. And that indeed is also, now we know today, is a major risk factor for transmission of Ebola. And what it means really is that in terms of transmission, it's close contact with body fluids, with a person infected with Ebola, a person or a dead body, because there's enormous viral load, virus excretion at death and for another 2–3 days. And what we found then also is that what we call case fatality rate, this is the mortality basically, was 100% among people who were infected through injection, and in terms of what we call person-to-person contact, and that was like more than 2/3 was only there, 83%. And that's what you would expect, because in most viral illnesses, if not all that I know, the outcome is, among other things, in function of how much virus is there in your body. If that's a high number, I mean a high viral load, then the outcome is usually very bad. And so we know today that the modes of transmission, they're still the same. There has not been any change. There's no airborne transmission and whatever. And that it correlates with the presence of a virus. It's an RNA virus. And here that you can detect that in, in serum of patients. And one of the challenges that we have is that it's very, very hard, very unusual to isolate the virus from someone who is already infected, but is not yet sick. And that would be very important to have a test like that, because if we could find out that, even 1 day after infection that we can already diagnose, that would simplify life enormously in terms of identifying those who are infected and those who are not, and who went to a funeral, for example, or family members. But it's not only in blood, it's isolated from urine, from stool. This is very limited data we will have soon far better data because of this epidemic. Breast milk, semen, so there's sexual transmission also, saliva, and skin. But except for semen and breast milk, it's all in people who are actually in acute illness, and it is really for those who are recovered. Semen is one of the, for about 3 months, that where virus can still be recovered from. On the average, about 7 to 8% of people who had been in contact with a case became infected, and that's what we call an attack rate. In epidemiology, there are lots of military terms, surveillance, attack rate, case fatality rate, but that's the terminology. So, okay, so now some basic mathematics, and this is a formula that actually comes from Oxford, from two scientists who worked here when they developed this concept, for Roy Anderson and Robert May. This is in 92 from publication, and they try to have a basic model of the spread of infections, of pathogens in a community, in a population. And this is called the reproductive rate, R0 or not, anyway, it's a bit, equals a product of beta, which is the probability of transmission, times the number of contacts, times the duration of infectiousness. And let me say a few words about that. The probability of transmission in terms of Ebola is actually very low for the general population. You really need very close contact. That's good news. It's unlike for measles, the probability of transmission is more than 20. When you have someone with measles who would sit here and you're not vaccinated, that would nearly certainly give rise to a big epidemic. And we've seen what's been in the news today, the last few weeks, in Disneyland in the US, major outbreaks of measles, because some people are reluctant to immunize their children, and a few of these cases have now serious complications of measles and encephalitis and so on, and I think there's even one death. So anyway, measles, 20, influenza also a lot, and norovirus is huge. If that happens in your hospital or in a daycare center or in any institution, that's a disaster. The number of contacts, C. Again, in Ebola, it's fairly limited. It's the people who care for a patient or who go to a funeral. Now, you can have super spreading effects. For example, one of the big events in this epidemic in Guinea was a woman who was a famous traditional healer, and when she died from Ebola, hundreds and hundreds of people came to her funeral and touched the body, because every culture has a way of saying farewell to the death. And in every culture, we clean the body, we wash it and so on. But then in Western and Central Africa, there is also really very close contact with the death, and the washing and so on is done with bare hands. So number of contacts is fairly limited, and that can also be very different if is the probability of transmission is high and you have mass events, yes, number of contacts, and then the duration of infectiousness. Here again, with Ebola, after infection, it takes about a week before you develop disease, and then a week later you're dead. I mean, that's basically the pattern. So not that much time, not long time to infect others. This is very different from example from HIV. HIV infection, you can be, even without treatment, be healthy for up to 10 years. And if you're very promiscuous, you can have many sex partners, and you can infect lots of people. And so I'm showing this because it gives you a really very good framework to think about any infectious disease, any epidemic. And in theory, Ebola is easy to contain. And that is what has happened for many years. But no longer now, as we know. And then the other question is, where is this virus coming from? Because we, we are a lousy host for Ebola virus. Because from the perspective of a virus, what's the purpose in life? This is the existential question. Purpose of life of a virus? It's have eternal life, to always find a host, because a virus cannot survive without a living cell. And, so it is, we're an accidental host. A host that is killed by the virus is not interesting, because then the virus has to jump to another host. So the ideal host is a host where there's nice symbiosis, nice relationship between the host and the virus. And in the beginning, monkeys were accused. This is a dried monkey from the market, not far from Yambuku. I went there back about a year ago for my 65th birthday, just probably the only tourist in the whole area. But my wife, we went to have a look, and we found there is bush meat. And, but I wouldn't be so condescending about that. I mean, here we eat deer, we eat all kinds of animals also, and it's just you go for the proteins that, are available. So for me, it's all a matter of taste. And also, you have bats. And the answer to where is this virus hiding is most probably is a kind of bat, fruit-eating bats. You've got basically two types of bats, insect-eating bats and fruit-eating bats, so vegetarians and non-vegetarians, basically. Bats have a very interesting immune system. They are carriers of many different viruses and don't feel a problem. They live quite in harmony with them. The problem is that the virus has never been isolated from a bat, from another virus. Only RNA has been detected, so that's some research. So there have been about 25 outbreaks between 1976, here, so the second known outbreak was in 94, then 95, and you see. And this is the number of cases, and always quite limited to a few hundreds of cases at the most in terms of deaths and cases. Everything changed about a year ago. I'm sorry. 12 months ago, there was, thank you. Yeah, I have some respiratory virus, and I don't know what it is. I've been coughing for, it's the 4th week. No fever. And a non-productive cough. And everything changed because in March last year, it was announced that Ebola was diagnosed in Guinea. Guinea is a country in West Africa, here, and is bordering Sierra Leone and Liberia and some other countries. And up to then, the common sense and the dogma was that Ebola is easy to contain. You isolate people, you treat them as much as you can, you put contacts in quarantine, make burials, funerals safe, and that's it, and it works. And that was the case until last year. And since then, today we have, and also I should say, all outbreaks up to now, with one exception in Ivory Coast, for one particular virus, and only one person who survived, everything else, all outbreaks were in Central Africa and East Central Africa, mostly in Congo, some in Gabon and Congo Brazzaville, and in South Sudan and Uganda. So I would understand that it took 3 months before the diagnosis of Ebola was made, because you can only find what you're looking for. And nobody was looking for Ebola in West Africa. And that's a big lesson. Never assume that something is not there. In the meantime, as I mentioned, we are around 23,000 cases, nearly 10,000 deaths. And all these cases come from one person. It's quite mind-blowing. The first case we know today was a there were 2 cases where brother and sister in Guinea, in an area near the border with Liberia and Sierra Leone, and that happened in December 2013. So that's 14, 15 months ago. And then it took 3 months to diagnose it, and then it took another 5 months, 6 months until August that this was declared as a public health emergency when there were already over 1,000 deaths. And so this is the epidemic curve, and we are now about here. So we are clearly over the peak of the epidemic, but it's not over regardless of what you may see, what you may read. First of all, in Liberia, indeed, over the last 2 weeks, they have not had one single new case. So that's good news, and it's really a strong indication that, the epidemic may be under control. The problem, though, is that borders are very theoretical, certainly for viruses, but also for people. It's the same families have a relative on either side of the border and so on, and the roads are good. But in Liberia had a pretty bad epidemic, and now it's really getting much better. Here, in contrast to Guinea, the number of cases that is also going down, but now went up again. And the same thing is true in Sierra Leone. So at the moment, this is figures up to the 8th of February, this is a month old, but we are at about 10,000 deaths and as I mentioned, 500 healthcare workers. This was from the situation on the 18th of February, and you can see that in darker blue where there are still new cases happening, particularly here in the forest areas of Guinea and of the western part of Sierra Leone. But in Liberia, things are much, much better. So why? Why was this epidemic different from all other epidemics? And I call it, this is an editorial I published in Science, and I call it a perfect storm. It's not just one explanation. It's unlikely that it's a new virus, a different virus that is more virulent or spreads faster. It is really a combination of things. One, very mobile populations. So the roads are fairly good, and people come across the borders and all that. And so you have a funeral in Oxford, and then cases pop up in Aberdeen in Scotland or so a week later. I mean, people just move. That's one thing. Two of the countries, Liberia, Sierra Leone, come out of civil war, very brutal civil war. You have blood diamonds. You may have seen the movies on that, and so there is no trust in what government says, and very divided societies. Healthcare systems are very, very defunct. Liberia had 51 medical doctors registered in 2010 for the whole country, for about 5 million people. So let's say one doctor or less than a doctor per 100,000 people. Just imagine. You can't do miracles with that. And above all was a slow response, denial in the beginning from the national authorities, from the international community. It was really MSF, Médecins Sans Frontières, Doctors Without Borders, who were really at the forefront of the fight against Ebola and trying to alert the world. Things have fortunately improved, and as of September particularly, and there are results also declining cases. Interestingly enough, in today's world, globalized world, an epidemic doesn't necessarily remain a local issue. Cases in the US, we had in London, in UK, we have in Portland, in Spain, and so on. So that's what happened also with other epidemics, and the most likely epidemic that will happen is influenza, and which spreads very easily over the world. So nearly at the end, what can we do? What's the strategy? And there's certain things we know, and that is isolation of patients so that there is no contact with others. The risk then is for the caregivers, and for example, MSF has lost, I think, 14, 15 colleagues. These things are really, it's very, very risky to to care for someone with Ebola. So we've really made enormous progress in terms of control of hospital transmission and also safe and dignified burials. Why am I putting dignified here? You have to give an opportunity to people to say farewell to their loved ones. And if you just put someone who died in a body bag and then that comes out, that creates lots of problems. People go underground, they're hiding their cases or their patients with the Ebola and so on. So that is now also something that I think we're doing much better now. Where it's not so clear is how to control transmission in community. We are in the age of Twitter, as you know, and so on, and there are the social media, and there are lots of rumors. Lots of rumors going on with mobile phone and all that, making it quite difficult. Many rumors say that, oh, these white doctors, they come and they spread Ebola. It's an American invention. It's the CIA, et cetera, et cetera. So dealing with that is really important. And it's not just rumors, but also in Guinea particularly, workers from the Red Cross, from WHO, from Médecins Sans Frontières have been attacked. Some have been killed, of villagers who thought that they're the ones who are spreading it. So it is something that you really need to take seriously and try to control. And it's the understanding the beliefs and the behaviors and so on. So that's what you see, huge, treatment units, very intensive, labor intensive, and then also making sure there is a clear messaging. And some of the messaging is not very helpful. This was from Angola, but it said that there is no cure for Ebola. And then on the other hand, they say, come to the hospital for treatment. Now, people are not stupid. And on the one hand, you say there's no cure, and then you say, why would you go to the hospital? So we have to really work all together, not only physicians and so on, but people who understand the culture and communication. So this is near the end. It's not over, but it's much better. And there are now about 15 to 20 new cases a day in Sierra Leone and Guinea combined. Many people now think that's fine. Normally, that's a reason for panic when you have that big alert. So we get used to things that we shouldn't get used to. And here, challenges are community engagement, so making sure that people are on board, that they believe it, that there's an increase in what they call security incidents, particularly in Guinea, continuing unsafe burials. Changing the way you say farewell to the death in any culture is extremely important. It's very, very difficult. We have a slight increase again, ongoing high case fatality rate, and slow progress in research, I should say. And just a few words about research. Also, because Oxford plays such a prominent role in it, we have, on the one hand, therapeutic and care research. How can we bring down mortality? On the one hand, by supportive care. I won't go into details, but also trying to use fairly primitive ways of treatment, and that is to use the plasma, the blood plasma of survivors, and to treat people with Ebola. And that's based on experience with hemorrhagic fevers in Latin America, Argentine hemorrhagic fever, where it was shown in a randomized trial compared to placebo that if you give plasma of a survivor, that reduces the mortality of someone with Argentine hemorrhagic fever, because that plasma contains very high levels of neutralizing antibodies. Is the same thing true for Ebola or not? We don't know, and there are studies going on at the moment. We have monoclonal antibodies that was in the news, so-called ZMapp, and we have a number of small molecules, and a team led by Dr. Peter Horby here from Oxford are conducting some trials here. And then we have vaccines also, where Professor Adrian Hill and his team, and we had also our previous speaker, doing some phase 1 trials, so seeing whether there are what the antibody response is and whether they're safe. the side effects are, and there are basically 3 vaccines that are being tested. 2 of them in West Africa already, one that comes from, that's produced by JSK. One that's also the tests have started recently in Sierra Leone, and now probably this week, also next week in Guinea. That's another vaccine that's based on a live vector. Sorry. And then another one that's produced by Johnson & Johnson and is a prime boost, and that we're involved with in Sierra Leone. It's a challenge to demonstrate whether these vaccines protect or not, because the number of cases is declining, and so that's the scientific challenge. But the alternative of not trying to evaluate it is not an option. Otherwise, by the next epidemic, we go back to square one. Just to end, Ebola is now in the news, but let's not forget that in just in the last 10 years, basically, these are just covers of Newsweek and Time, but SARS, then bird flu, then another flu type. We had MERS in the Middle East, bird flu, and now Ebola. So even if infectious diseases are on a decline, and my professors were right that there was less of a problem, but they were wrong when they say there was no future in it, that there will always be emerging infections. And we need to be prepared as a world, as a country, including with the facilities to develop much faster vaccines and so on. So this is the end. I wrote some of that you can read. This is a commercial, in my book called No Time to Lose. Thank you very much.